Hello students, welcome to Bridge Academy for Grade 10 Biology Tutorials, Unit 1, Lesson 3. Major discoveries that revolutionized biology. At the end of this lesson, you will be able to discuss at least 5 major discoveries that revolutionized biology. Introduction. Biology is the study of life, from tiny microbes to complex animals. Over centuries, scientists have made discoveries that changed how we understand living things. Let us move from ancient ideas to modern breakthroughs, showing how each discovery built on the past. Aristotle was a Greek philosopher who lived from 384 to 320 BC. He loved observing nature. Aristotle was one of the first to study living things systematically. He wanted to understand how plants, animals, and objects were related. Aristotle created a system called the ladder of life. This system grouped living things into broad categories called genera, like mammals, birds, or fish. Within each genus, he named specific species, such as lions or eagles. Aristotle's system was the first to organize life based on shared traits. For example, he noticed mammals have hair and nurse their young. His ideas laid the foundation for taxonomy, the science of classifying organisms. Aristotle's work influenced later scientists, like Carl Linnaeus, who built a better classification system. His discovery was the starting point for organizing life's diversity. 2. Galen 129 to 161 AD. Galen, a doctor in ancient Rome, studied the human body by dissecting animals like pigs and monkeys, as human dissection was forbidden. He learned how organs like the heart, lungs, and nerves work together. Galen wrote detailed books about anatomy, blood flow, and the nervous system. His ideas guided doctors for over 1,000 years. However, some ideas were wrong because he studied animals, not humans. Despite this, Galen's careful observations and writings helped future doctors understand anatomy, setting the stage for modern medicine. 3. Antony van Leeuwenhoek 163 to 173. Antony van Leeuwenhoek, a Dutch shopkeeper, had a passion for making lenses. He crafted simple but powerful microscopes that magnified up to 270 times, far better than others in his time. Using these, he was the first to see tiny living things like bacteria, yeast, and sperm cells, which he called animalcules. He observed these in water, saliva, and other samples. His discoveries revealed a hidden world of microscopic life, sparking new studies in microbiology and showing that life exists in forms invisible to the naked eye. 4. Carl Linnaeus 1707-1775. Binomial nomenclature. Carl Linnaeus was a Swedish scientist who lived from 1707 to 1775. He made it easier to name and study living things. Linnaeus invented binomial nomenclature, a system that gives each species a 2-part Latin name. The first part is the genus, and the second is the specific epithet. For example, humans are Homo sapiens. Homo is the genus, and sapiens is the specific epithet. Linnaeus also created a hierarchical system to classify life. He grouped organisms into kingdoms, classes, orders, families, genera, and species. This system helped scientists study biodiversity. Linnaeus wrote a book called Systematiae, which listed many plants and animals. His work made it possible for scientists worldwide to share knowledge about species. Linnaeus's system is still used today. 5. Charles Darwin and evolution, 1809 to 1884. Charles Darwin was an English scientist who lived from 1809 to 1882. He changed biology with his theory of evolution. Darwin said species change over time through natural selection. In natural selection, organisms with helpful traits survive better and reproduce. These traits pass to their offspring, leading to new species over time. Darwin published his ideas in 1859 in On the Origin of Species. He used evidence from fossils, animal locations, and studies of plants and animals. Darwin also said all living things share a common ancestor. This idea, called descent with modification, explains life's diversity. Darwin's work built on Linnaeus's classification by showing why species are different. 6. Gregor Mendel and Genetics, 1822 to 1884. Gregor Mendel was an Austrian monk who lived from 1822 to 1884. He is called the father of genetics. Mendel studied how traits pass from parents to offspring. He used pea plants because they grow fast and have clear traits like seed color or plant height. Mendel crossed plants with different traits and tracked the results. Mendel discovered 3 laws of inheritance. The law of segregation says each parent gives 1 copy of a trait to their offspring. The law of independent assortment says traits are inherited separately. Mendel's big discoveries, the 3 laws of inheritance. Mendel's experiments led to 3 key ideas, now called his laws of inheritance. Let's break them down. Law of segregation. Every living thing gets 2 copies of a trait, 1 from each parent. For example, if we're talking about seed color, a plant gets 1 color instruction from its mom and 1 from its dad. When it's time to make seeds, those instructions split up and each seed gets just 1 copy. Think of it like flipping a coin. Each kid gets a random pick from mom or dad's traits. Law of independent assortment. Different traits don't stick together, they're passed on independently. For instance, whether a pea plant is tall or short doesn't affect whether its seeds are green or yellow. It's like rolling dice for each trait separately. This explains why you can have a tall plant with yellow seeds or a short plant with green seeds. Law of dominance. Some traits are stronger than others. If a plant inherits a strong dominant trait, like purple flowers, it'll show up and hide the weaker recessive trait, like white flowers. But the recessive trait is still there, just hidden, and can pop up in later generations if paired with another recessive trait. 7. Louis Pasteur and the Germ Theory, 1822 to 1895. Louis Pasteur was a French scientist who lived from 1822 to 1895. He transformed biology with the germ theory of disease. Before Pasteur, people thought diseases came from bad air. Pasteur showed that microorganisms, like bacteria, cause diseases such as anthrax and cholera. His experiments proved that germs spread illness. Pasteur also invented pasteurization. This process heats food or drinks, like milk, to kill harmful bacteria, then cools them quickly. Pasteur developed vaccines by weakening microbes. His vaccines for anthrax and rabies saved many lives. Pasteur's work connected to Mendel's by showing how microbes affect health, influencing which traits help organisms survive. 8. Robert Koch 1-8 for 31910. Bacteriology. Robert Koch was a German scientist who lived from 1843 to 1910. He is known as the father of bacteriology. Koch built on Pasteur's germ theory. He developed ways to find specific bacteria that cause diseases. Koch created Koch's postulates, a set of 4 rules to prove a bacterium causes a disease. The rules are: I find the germ in sick organisms. 2. Grow the germ in a lab. 3. Infect a healthy organism with the germ to cause the same disease. 4. Find the same germ in the newly sick organism. Koch's work in the late 1800s improved disease identification and treatment, saving millions of lives. Koch used these rules to identify bacteria causing tuberculosis, cholera, and anthrax. He also improved lab techniques like growing bacteria on agar in petri dishes. Koch's work made Pasteur's germ theory practical for medicine. His discoveries helped explain how diseases affect survival, linking to Mendel's ideas about inherited traits. 9. Jane Goodall and primate behavior, 1934. 9. Jane Goodall and primate behavior, 1934. She discovered they make and use tools like sticks to get termites. This showed animals have intelligence, challenging the idea that only humans use tools. Goodall also observed that chimpanzees have complex social behaviors. They form groups, show emotions, and have unique personalities. Her work highlighted the need for conservation as habitat loss harms chimpanzees. Goodall's findings connected to Darwin's evolution by showing how behavior affects survival. They also linked to Mendel's genetics, as behaviors may be influenced by inherited traits. 10. Barbara McClintock 1902-1992. Barbara McClintock, an American scientist, studied corn plants in the 1940s and 1950s. She discovered that some genes can jump or move within a plant's DNA, changing traits like kernel color. These jumping genes, called transposons, showed that DNA can rearrange itself, affecting how traits are expressed. Her ideas were doubted at first, but earned her a Nobel Prize in 1983. McClintock's work revealed how flexible DNA is, influencing genetics and our understanding of evolution. 11. James Watson 1928 and Francis Crick 1916-2004. James Watson and Francis Crick were scientists who worked together in the 1950s. In 1953, they discovered the structure of DNA, the molecule that carries genetic information. They found that DNA has a double helix shape, like a twisted ladder. The sides are made of sugar and phosphate, and the rungs are for bases: adenine, thymine, guanine, and cytosine. These bases pair in specific ways. Their discovery showed how DNA stores and copies genetic information. It explained how traits are passed during cell division. Watson and Crick used X-ray images from Rosalind Franklin and data from Morris Wilkins. Their work won a Nobel Prize in 1962. The DNA discovery connected Mendel's genetics to molecular biology, showing how traits are coded in genes. 12. Ian Wilmot 1994 and Keith Campbell 1944 and 1954-2012. Ian Wilmot and Keith Campbell, British scientists, achieved a breakthrough in 1996 by cloning a sheep named Dolly at the Roslin Institute in Scotland. Cloning creates an exact genetic copy of an organism. They took DNA from an adult sheep's cell, placed it in an empty egg cell, and grew it into a new sheep. Dolly was the first mammal cloned from an adult cell. Their work showed that adult cells can be reprogrammed, opening possibilities for medical research, like growing organs, but also sparking debates about cloning ethics. Summary: the groundbreaking work of 8 scientists revolutionized biology, shaping our understanding of life. Aristotle introduced the ladder of life, the first system to classify organisms into genera and species, laying the foundation for taxonomy. Carl Linnaeus developed binomial nomenclature, standardizing species naming with 2-part Latin names, and created a hierarchical classification system. Charles Darwin proposed the theory of evolution through natural selection, explaining species diversity via descent with modification. Gregor Mendel established the laws of inheritance, founding genetics by showing how traits pass through generations. Louis Pasteur proved the germ theory, developed pasteurization, and pioneered vaccines, transforming medicine. Robert Koch advanced bacteriology with Koch's postulates, identifying disease-causing bacteria like tuberculosis. James Watson and Francis Crick discovered DNA's double helix structure, launching molecular biology and explaining genetic inheritance. Jane Goodall revealed chimpanzees' tool use and social behaviors, emphasizing conservation and animal intelligence. Their interconnected discoveries form the backbone of modern biology, enabling research from genes to ecosystems. Review questions and answers. 1. What was Aristotle's ladder of life, and why was it significant? Answer: the ladder of life was Aristotle's system to classify living things into genera and species based on shared traits. It was significant as the first organized classification, influencing modern taxonomy. 2. How did Linnaeus's binomial nomenclature improve biology? Answer. Binomial nomenclature gives each species a two-part Latin name, genus, and specific epithet. It's standardized naming, making it easier for scientists to communicate and study species globally. 3. According to Darwin, what is natural selection? Answer. Natural selection is when organisms with traits that help them survive are more likely to reproduce, passing those traits to offspring, leading to species changes over time. 4. How did Mendel's laws of inheritance contribute to genetics? Answer. Mendel's law's segregation, independent assortment, dominance explained how traits are passed from parents to offspring, forming the basis of genetics and supporting evolution. 5. How did Watson and Crick's DNA discovery connect to earlier discoveries? Answer. Watson and Crick's discovery of DNA's double helix showed how genetic information is stored and copied, explaining Mendel's inheritance laws at the molecular level and supporting Darwin's evolution. Thank you. Do not forget to like, share, and subscribe.